Hey guys, welcome back. This is Lei. In this video, I'm gonna show you how to predict cancer subtypes using RNA-seq data. The methods I'm gonna show you are actually not limited to cancers. They can be applied to any other molecular subtyping scenarios as long as you have the gene signatures. Here I use breast cancer subtypes as an example. The heat map shows expression of genes in the PAM50 gene signature, which classified about 1,900 patients into 5 breast cancer subtypes. The TDA signature further classified these patients. So what is molecular subtyping? It's a classification of cancers based on expression patterns of gene signatures. The gene signatures here refer to sets of specific genes whose combined expression patterns serve as biomarkers to these subtypes. Remember the pathway enrichment analysis. Each pathway is characterized by a set of genes. This set of genes is known as a gene signature. Here are some example gene signatures for breast cancer subtypes. Among them, PAM50 is the gold standard gene signature for breast cancer subtyping. So we're gonna do subtyping in R. So our first step is to install the R packages we need, as shown here. Among them, Genefu and PAM50 are the packages specifically designed for breast cancer subtyping. Well, GSVA can be used to any molecular subtyping. Once installed, we load the packages in our R Studio. And our next step is to prepare the data. Here I use a package called GeoQuery to download the example data from the NCBI Geo database. I use the getGeo function to download the sample information table, and I download the counts table directly from the website and read it in using the fread function. So the tables are in the following format. The top table is the sample information table. The bottom one is gene expression table. So make sure the sample IDs are consistent between the two tables. Once we have the tables, we can process them in the same way as we did in the differential expression tutorial. So basically, QC and the normalization and the subsetting. So we're interested in breast cancer, so we only need the breast samples and combine the samples into a DGE list. Remember, the object has both expression table and sample table in it. Because some subtyping packages require gene entries ID instead of gene symbols, so we have to make a gene annotation table using the following code. So this code will generate a gene annotation table in this format. So this last column is gene entries ID. So make sure the genes and the order match the ones in our DGE gene expression matrix. So ignore the last part, which I used to subset the dataset for demonstration purpose. So now that we have the data prepared, we can perform breast cancer subtype classification using the PAM50 package. So the PAM50 package requires a gene expression matrix labeled with gene entries ID. So we just need to retrieve the gene expression matrix from our DGE list and rename all the genes using the entries ID. Once you have it, fit it in the PAM50 function and call the function. That's it. And the results will be stored in a table in this format. So the numbers represent the predicting probability. The first column represents the final predictions. and each row represents a sample. So basically, the final prediction is based on the probability, the largest probability. For example, the first sample, the basal has a 99.99% probability. So we just call it the basal. And the second one, Luminal B, 99.8%. And you can save the table to your computer using the fwrite function. You can also plot the gene expression of the PAM50 signature in our dataset using the plot PAM50 function, and save the heat map in this format. So we can also predict cancer subtypes using the Genefu function. The Genefu package. The main predicting function in the Genefu package is the molecular subtyping function, which requires 3 inputs. The subtype model option, which allows different gene signatures. Here we still use the PAM50 signature. And here are some built-in signatures we can choose. The data option requires a transposed gene expression matrix. So we transpose our gene expression matrix using the transpose function T. And an gene annotation table we just created. Once we have all 3 inputs, we put them into the molecular subtyping function and execute the function. And then we retrieve the prediction table the same way, and it had the same format. Numbers are the predicting probability, and last column contains the final predictions. We can also plot the predicting probability using the heat map, p-heatmap function, and save the results to our computer using the PDF function over here. So both method 1 and method 2 are designed for breast cancer subtyping. So, but what if you are working on other cancers? You can search if there are already existing packages for your cancer types. But if you don't, you can use the GSVA package as long as you have the gene signatures. For example, in our case, we have 4 customized gene signatures: Basal 1, Basal 2, Luminal 1, Luminal 2. We put all 4 gene signatures in a list, and we use the single cell GSVA method to predict our subtypes. So this function accepts 2 options, 2 inputs. One is the gene expression matrix. The other one is gene signature. So we can use the gene symbols now. So it doesn't need gene entries ID. So we set our parameters this way and then call the GSVA function to predict the results. If you want to compare the prediction across a large amount of samples in a large cohort, you can use GSVA method. But for now, we want a single sample profiling. So we use the single sample GSVA method. So the resulted table has similar format, but the numbers are no longer predicting probability anymore. It's enrichment scores, just like our GSVA pathway enrichment analysis. So just pick up the highest score as our final prediction. You can also plot the enrichment scores in the heat map using the p-heatmap function in the same way. So these are all 3 methods for cancer subtype prediction. And you can go to my website and copy the code there and try it on your own. And in the next video, I'm going to show you how to do deconvolution analysis for RNA-seq dataset.